Hello everyone, welcome to another session. Today we will be discussing about the fate of an alien piece of DNA when introduced into a host organism, and we will be discussing about the historic experiment, the first instance of construction of an artificial recombinant DNA molecule within a laboratory. But before getting into the new topic, I would like to go for a short recap of what we have learnt in the previous class. So in the previous class, we tried to understand what is the definition of biotechnology, and we also tried to find out what are the two core areas of biotechnology. You have seen that the two core areas of biotechnology are genetic engineering and maintenance of sterile culture medium. Genetic engineering and maintenance of sterile optimum environment for the growth of recombinant cells. So one part is essentially involved with designing of the recombinant DNA, and the second part is essentially involved with the large scale production of the given cell, right? So biotechnology is essentially a production process which is happening at a large scale, and during the production process we are growing, we are cultivating some cells or some biological material which will give us the product, and this product should definitely be useful to uplift the human standards, right? So after looking into the two core areas of biotechnology, we tried to learn about how the recombinant DNA technology is different or is superior to sexual reproduction. Why? Because in sexual reproduction and in recombinant DNA technology, recombination is a common process. Recombination of DNA is a common process in both the cases. Then, how is the recombinant DNA technology much superior when compared to sexual reproduction? Though sexual reproduction allows variations, it has certain limitations. What are those limitations? It cannot cross species barrier. And the second thing is, when you go to perform a traditional hybridization experiment, the traditional hybrids can receive both desired genes as well as undesired genes. That means the chances of the progeny after a hybridization experiment receiving both desired genes and undesired genes is equal, and for this reason this is the major drawback of traditional hybridization, which involves sexual reproduction. But in recombinant DNA technology, we are picking the genes of our choice. We are picking the genes of our choice pinpointedly, and only desired genes are introduced. Only desired genes introduced. And this is how the drawback of the traditional hybridization is overcome with the help of recombinant DNA technology, right? Now, let us see what is the fate of an alien piece of DNA when introduced into the host organism. And before that, one more special feature I would like to tell you is that the kind of gene transfer that takes place from parent to offspring in natural sexual reproduction, the kind of gene transfer that takes place from parent to offspring is a kind called as vertical gene transfer. Whereas in recombinant DNA technology, the gene transfer that happens between two different organisms is called as horizontal gene transfer. That is, the direction in which the genes are flowing in the natural sexual reproduction is vertical from parent to offspring, whereas the direction of gene flow in recombinant DNA technology is from one organism to another organism. This is referred to as horizontal gene transfer. Even horizontal gene transfer is not a completely new process. It takes place within some bacterial cells. Now, if we get into the details of an alien piece of DNA, if you can see here, this is the donor DNA, and the donor DNA is trying to enter into the recipient cell. So here the recipient cell can be called as a host cell. So whenever a piece of DNA which is foreign, non-self, is otherwise called as alien DNA. A foreign DNA, non-self DNA is otherwise called as alien DNA. When an alien piece of DNA enters into the host cell, what is going to happen to that? That is what we are going to see now. So as you can see here, it was somehow able to enter into the host organism. But after its entry, there are four major possibilities. Number one, as soon as it enters, the host will have certain enzymes called restriction enzymes, and these enzymes will digest the foreign DNA into fragments. That is, as soon as a foreign piece of DNA enters into a bacterial cell, the bacterial enzymes will break the foreign DNA into bits, and that process is called as restriction digestion. Right? And in the second case, this is the first case, and in the second case, the donor DNA is somehow able to withstand the bacterial enzymes. That means within the bacterium, the donor DNA was able to protect itself from the restriction enzymes. In that case, the donor will stay here like this in a passive mode. It can neither replicate nor be transferred into the progeny cells. This is the second case, that is, and the third case is if the donor DNA is a plasmid. If the donor DNA is a plasmid. What is a plasmid? Plasmid is a self-replicating piece of DNA. A plasmid is a circular DNA molecule which can replicate on its own. And if the donor DNA happens to be a plasmid, it can also stay in the host cell stably. And the fourth case, if the piece of donor DNA is able to integrate in the host genome, if it is able to integrate in the host chromosome, even in this case, it can be stably present in the host organism. So as you can see here, in case 1 and case 2, there is no scope for the donor DNA to be propagated into the cells. That means even this cell undergoes cell division, you don't find the donor DNA here. There is no donor DNA in the progeny. This is the progeny. Even here, though it was able to withstand the restriction digestion, the progeny cells, it cannot be transferred into the progeny cells. In progeny, there is no donor DNA. So in the second case also, even though the donor was able to withstand the restriction enzymes, it cannot be transferred to the progeny. So no donor in the progeny. Only in the third case and the fourth case, if the DNA happened to be a plasmid or if the DNA was able to integrate into the host chromosome, only in these two cases, the progeny cells can receive the plasmid DNA along with the chromosomal DNA. This is the chromosomal DNA and this is the plasmid DNA. Okay. Now, if I ask you what makes the difference, why the plasmid DNA was able to be stably transferred into the progeny and why the piece of DNA when it is become part of the host chromosome is able to stably transfer into the progeny. The answer is, when the donor DNA is part of the chromosome or when it is part of the plasmid, it is having a specific sequence called as origin of replication. That means whenever a piece of DNA needs to replicate, it needs a special sequence called as origin of replication. And origin of replication will help in initiating the replication process. So always remember, a piece of DNA is capable of replication only when it is having a specific sequence of DNA called as origin of replication. And in this case, the chromosome is having an origin of replication, plasmid is also having an origin of replication. And for this reason, whenever the donor piece of DNA is linked with chromosome or a plasmid, it can be replicated and it can stably be transferred into the progeny cells, right? So this is the fate of an alien piece of DNA when introduced into a host organism. Now, let us look at the first instance of construction of artificial DNA. There are two scientists involved in this. It is Herbert Boyer and Stanley Cohen. And this is Herbert Boyer and this is Stanley Cohen. As you can see, they have accomplished this task in the year 1972. Very little was known when they did this experiment. So here, Boyer was an assistant professor in the University of California. He was working with a set of enzymes called as restriction enzymes. These restriction enzymes are present in the E. coli bacterium. What was the significance of these restriction enzymes? Boyer observed that these restriction enzymes are making cuts in a DNA, not at random, but at a specific sequence. And as a result, the fragments generated after the cut are having a specific pattern. As you can see here, if this is the piece of DNA that is cut by the restriction enzyme, so in general, if we take the piece of DNA as a ribbon like this, if we make a cut, what will happen? Two fragments will be generated like this, right? But here, that is not the case. The restriction enzyme that is cutting the DNA into two fragments is generating two different kinds of, it is able to generate two fragments which are having single-stranded overhangs. Two fragments which are having single-stranded overhangs like this. And such fragments are called as sticky-ended fragments. As you can see here, when the DNA is cut with a restriction enzyme called EcoR1, this is the name of the restriction enzyme. This enzyme is making a cut in such a way that each fragment is having a single-stranded overhang, right? Now, such fragments which are having a single-stranded overhangs are otherwise called as sticky-ended fragments. What is their importance in rDNA technology? If you can see, observe carefully, the sticky-ended fragments generated by the same enzyme they are complementary to each other. So this is TATA and this is AATT. TTAA is complementary to AATT, right? So in this case, whenever sticky-ended fragments are generated from a single enzyme, both the fragments can be easily joined in this case. And this is how restriction enzymes are more useful. Now coming to Stanley Cohen. Stanley Cohen is from the University of Stanford. He was working with ringlets of DNA called as plasmids, right? and these ringlets of DNA were found freely floating in the cytoplasm. So here, if you take a cell, a bacterial cell, it has a chromosomal DNA. Apart from this, it is also having a plasmid DNA, and this plasmid DNA is called as the ringlet DNA. What is the speciality of this plasmid DNA? A plasmid DNA is a small circular DNA which is having its own origin of replication. And as a result, a plasmid is capable of replicating on its own without depending on the host chromosome, right? Now, let us see what Stanley did with these plasmids. He developed a method by which the plasmid can be extracted from one cell and it can be placed into another cell. Literally, he was playing, he was trying to do some kind of experimentation. So initially he observed the plasmid, he extracted it, that means he pulled it out from the cell, and then he reintroduced it into another host organism, right? Now, with the help of the techniques developed by Boyer and Cohen, let us see what they did. So, these two people, this Cohen and Boyer, they met at a particular conference and they were having a talk over a coffee table. So, what we say here, chai pe charcha hota hai na, वैसे. So, during their conversation, they came up with an interesting collaboration and they made an experiment. So, what was that experiment? Let us see. And in this experiment, they used the enzymes what they observed. Boyer used the enzyme what he observed, and Cohen tried to use the plasmid extraction method. So if you can see here, a plasmid from Salmonella typhimurium was extracted. So plasmid extraction was done by Cohen. A plasmid from Salmonella typhimurium, a type of bacterium, was extracted, and then another plasmid from some other unknown organism was also extracted. That means, using the methods of Cohen, two plasmids were extracted from their respective host cells. One is from Salmonella typhimurium and the other one is from an unknown bacterium, right? Now, after extracting the plasmids, they tried to cut the plasmids with restriction enzymes. So, cut the plasmid with restriction enzyme. Here and here, the same restriction enzyme was used. Now, who told us about the restriction enzyme? It was Boyer, right? So, using the Boyer's restriction enzyme knowledge, they cut both the plasmids, and after that, they joined these cut plasmid with the help of an enzyme called DNA ligase. And this is how they are able to generate a recombinant DNA molecule. So as you can see here, this plasmid is from one organism, this plasmid is from another organism. Now the second plasmid is a source of the desired gene. What is the desired gene here? The desired gene here is an antibiotic resistant gene, right? And we want to introduce the antibiotic resistant gene into a plasmid. Now, with the help of the restriction enzymes, after cutting the plasmid, we are able to link the desired DNA, that is antibiotic resistant gene, to the cut plasmid. That linkage ke liye, we took the help of another enzyme called DNA ligase. Now, once the recombinant plasmid has come, in the next step, we tried to, this is the recombinant plasmid, right? Now, this is the designing part. Now, once the recombinant plasmid is designed, it was introduced into another organism. What was that organism? It was E. coli. It was E. coli. So, here, now the DNA is from two different sources and it is introduced into a third organism. And in this way, E. coli was transformed with the help of gene transfer techniques. And as a result, the E. coli became antibiotic resistant. E. coli naturally does not contain any plasmid and it is not having any resistance to antibiotics. But after the plasmid containing antibiotic resistant gene is transferred into E. coli has become resistant to the antibiotic, right? So, This is the brief work what they have done. Isolation of plasmid DNA and isolation of desired DNA, splicing the DNA, that is chopping off the DNA, and recombining both the fragments of DNA, inserting it into the bacterial cells and making use of the bacterial cell to produce a particular product due to the desired genes. Right? So dear friends, it's a multi-step process to construct a product and to go for a large scale production. If you look into the two core areas of biotechnology, one is concerned with the design of a recombinant molecule and the second area is concerned with the large scale production of the recombinant molecule. So as I told you, it is a multi-step process and it takes help of so many tools and techniques. So if you look into the basic steps, how do we design it? How do we design a recombinant DNA? This is the first major area of biotechnology, and the second major area is to produce the designer DNA on a large scale, produce the recombinant product on a large scale. So as you can see in this diagram, these are the basic steps involved in designing a recombinant DNA molecule and putting it into the host cell. Look at the first step 1. You have to purify the DNA first of all. The purified DNA sample should be digested with restriction enzymes, and once the digestion is completed, the desired fragment of DNA should be inserted into the vector molecule. A vector DNA is otherwise called as a vehicle DNA molecule, which can carry the desired piece of DNA safely into the host cell, right? So what are we going to do now is, once we get the purified sample of DNA, we need to cut it with the restriction enzymes and we collect the piece of desired gene and insert it into the cut vector. Now this is how we have designed the recombinant DNA molecule. Now once the recombinant DNA is ready with us, we need to insert it into the host cell. A host cell is a cell into which we are going to insert the recombinant DNA molecule, and the process by which the recombinant DNA is taken up by the host cell is called as transformation. Now once the cell is transformed, we should provide conditions in such a way that the recombinant DNA should be propagated, it should be stably passed to the next generations, right? So this is how we have to design the recombinant DNA molecule and insert it into the host organism. Now that the host organism is ready, now you are calling the host which is carrying the recombinant DNA as the recombinant host. Now this is the designer product that we made here in the first step. Now in the next step, this recombinant host should be grown or cultivated on a large scale, right? And that includes chemical engineering processes and maintenance of the sterile culture medium, etc. So once the designer piece is ready, we are going to cultivate it on a large scale and we are going to harvest the product, purify it and sell it in the market. So basically friends, biotechnology is a process, it's an industrial process. So if we want to understand and if we want to get the flavor of biotechnology, we have to imagine ourselves as biotechnologists and try to get into the details. Now, if you look at the multi-step process, this is like a roadmap for you. If we want to become a genetic engineers, when we want to design a product and cultivate it on a large scale, as I told you, it's a multi-step process. We can make use of these steps as a roadmap to make the product. The very first step is going to be isolation and purification of the DNA sample. So this can be achieved with the help of cell disruption methods and purification methods. So once we get the purified form of DNA, we are going to digest it. We are going to cut the long DNA molecule into shorter fragments with the help of another enzyme called restriction enzymes. Since restriction enzymes are capable of cutting DNA or RNA, they are otherwise called as molecular scissors. So now the purified sample is chopped off with the help of restriction enzymes into shorter fragments. Once we get these shorter fragments, these fragments should be separated based on their size with the help of another technique called agarose gel electrophoresis, right? So the fragments should be separated based on their size in a technique called gel electrophoresis. Next, in the next step, after the cut fragments are taken, we are supposed to link them together with the help of another enzyme called as DNA ligase. Now DNA ligase, because it is capable of joining two DNA fragments, it is otherwise called as a molecular glue. Now we have the recombinant DNA molecule in our hand, the designer DNA. Now this designer DNA should be inserted into the host cell in a process called transformation. Right? These are the steps you can go through. So the next steps will be multiplication of the desired DNA, right? So once the designer DNA, the recombinant DNA entered into the host organism and the host was able to replicate it stably and the designer DNA was able to propagate itself into the next generations. That means stable transfer of the recombinant DNA is happening due to the presence of DNA polymerases. So what happens actually here is, as soon as the recombinant DNA molecule is carried into the host organism, within the host cell, the plasmid tries to replicate on its own with the help of the host enzymes, that is DNA polymerases, right? Now, the problem here is, once the transformation experiment was done, we cannot expect that all the host organisms have taken up the recombinant DNA molecule. And for this reason, we have to undergo the screening test. Screening test will enable us to select only those host cells which are carrying the recombinant DNA molecule. And then, once the screening is completed, now we have the recombinant host. Now that the host cell is having the recombinant DNA molecule, the host is otherwise called as a recombinant host. This is the designer product we have in our hand, and with this designer product we have to go for the large scale cultivation, and this process is called as scaling up. Now once the large scale cultivation is done, where you have to provide all the optimum conditions for the growth of recombinant host, as a last step we have to harvest the product in a step called downstream processing. So as you have seen here, the biotechnological process is a multi-step process which involves a total of 9 steps altogether, and at each step we have to make use of so many tools and techniques.